Alright, so what we have here is a video that's going to talk a little bit about protein expression and kind of give you a little bit more context of what the role of DNA is. Yeah, you've been learning about the structure of DNA and where the bonding happens and why it's important. Um, and so I'm going to kind of do a little bit of an overview, but this is meant to build on the notes that we took a couple days ago on transcription. So let's just review a little bit on what we know about the cell cycle. Again, pretend that's a nice circle. Um, it is not, but that's okay. And we know that when we're talking about the cell cycle, let's go and label it such. The cell cycle, we're talking about interphase particularly, and we're talking about G1, S, and G2. And we're going to come back to S, but not in this video, right? So that's going to be next. We're going to talk about how DNA makes copies of itself. But before we do that, we need to think about what the point of DNA is. And remember that DNA, the whole point of DNA is to be read. It'll be turned into RNA, most notably in our story to messenger RNA, which then gets read and then translated into proteins, which are made out of amino acids. So we're going to pull on some information you should already know. And I'm not going to really review it, but I expect you to be able to kind of synthesize it and put it together. So again, we're talking about what's happening really starting in G1 in this sequencing. And so this first part is transcription, and the second part is translation. And I'm not going to really pick up on a lot of detail on what's going in transcription, so you should review your notes to review the role of RNA polymerase and how base pairs work. But I'm just going to kind of go through this kind of quickly. So let's say that we've got, I'm going to do my sugar phosphate backbone in this kind of boring gray, because that's not really the point of the DNA, right? The sugar phosphate backbone, the sides of the nucleotide are really they're kind of a structure to kind of create this double helix and allow the nitrogen bases to pair up, right? So we get these nitrogen base pairs all the way across this DNA strand, and we need to know that we're working with complementary base pairs, all right? Complementary base pairs. So adenine goes to thymine, cytosine to guanine in DNA, and this is our double-stranded DNA. So we should also know that we have deoxyribose sugars. But what ends up happening is, I'm going to see if I can erase some of this here. This DNA strand actually opens up. So let's go ahead and I'm going to erase this part. You might want to redraw your little double helix there. And what we know is, oops, forgot to go back to my pen tool, is that there's an enzyme RNA polymerase that comes through, unwraps it, and it will add in new nucleotides. So it might add in new nucleotides one at a time to the new strand, right? So this yellow strand is mRNA. And you should know that the way that this pairing works, if it's DNA, we're talking about DNA, I'm going to do this in this boring gray, that A and T go together, and C and G go together. And that you should know because that's kind of how you're doing your pairings for your modeling. But when we're talking about DNA to RNA, okay, so DNA to RNA, we have to think about this a little differently. All right, so this is involved in transcription, right? So DNA, to RNA. So let's do cytosine, guanine, thymine, and adenine. And then we need to think about what it would base pair to. Not what it gets turned into. That's not what this arrow means. It means is what is it pairing to, right? So when DNA is being transcribed by RNA polymerase, the base pair that matches it is going to be also complementary, okay? So this word also is relevant here. So C to G, G to C, T to A, and A to U, all right? So we've got uracil here. So if I give you a sequence with U's in it, then you know it's RNA. If it's got T's in it, it's DNA. And so that's going to be kind of the first step, in that this code that's on the DNA gets complemented to the mRNA. And in your notes before, we said that the mRNA leaves for the ribosomes. It's looking for ribosomes. And that's the key, right? So the ribosomes are going to be what's really, really important here. So what we're going to do is talk about this next step. So this is all transcription. And let's now talk about, and that's what we talked about in class. So what we're going to talk about now is the next step, which is the second part of all this, which is translation. So translation, if you're translating something, you're taking it from another language, right? So this is taking this code from nucleic acids in RNA, ribonucleic acid, and converting that into, which it would have ribose instead of deoxyribose, would go from nucleic acid to amino acids. Okay? And so I'm going to draw a diagram here, and it's going to get a little complicated, but what I'm going to do is draw my mRNA sequence, and each of these lines is a nitrogenous base. All right, and then what we're going to know, what we need to know, is that there are two parts to the ribosome. So we might say the ribosome, one part is here. We're going to call this the small subunit. And then there's a larger piece that is the large subunit. So there are two parts to a ribosome. So think about how weird it is that when we draw a cell, and we do our ribosomes as these little dots in the cell, these are really tiny. And so we know that these ribosomes are made out of something quite important. So this yellow thing is mRNA. The purple parts are made out of what we call ribosomal RNA, rRNA, and proteins. I'm going to show you a JMOL file of this in just a second. And you're welcome to play with it as well. I'll put it up in the folder that you can pull down and play with in JMOL too. So what ends up happening is that, and I'm going to give you a very, very simplified model, okay? It's much more complicated than this, but this is a very simplified model, so you need to know this story. So what ends up happening on this mRNA sequence is that there might be, we're going to write this as like AU, AUG, C, U, G, G, C, A, U, U, A. Okay, so it could keep going on. This user dead giveaway that we're talking about messenger RNA. And what ends up happening is that the ribosome reads things in terms of codons. Okay, codons are sets of 3 nucleotides, but really what it's reading are the nitrogen bases, because that's the only thing that has the code in a nucleotide, right? So the sugar and the phosphate are the same. So it's going to read these in sets of 3 nucleotides. So what I like to do is read this in terms of sets of three and then mark them. So I've got AUA, UGC, but there's one other little caveat, and I'm going to do this in red here, that the start codon for absolutely every single protein that we know of starts with the codon AUG, and if because of this U, you know we're talking about mRNA. Okay. You'll see a question here about what is the DNA sequence that led to AUG, and you should fill that in. But we're going to look for AUG first, and I see AU, no G, AUG. Okay, so here's my first codon. Right here, I'm going to draw a line, AUG, then go 1, 2, 3, draw a line, 1, 2, 3, draw a line, 1, 2, 3, draw a line, and you'd keep going. And so what ends up happening, not that there are actual lines drawn here, but what ends up happening is another molecule comes in and it pairs with this AUG. So I'm going to draw it kind of in a strange way because there are lots of models of this. This thing has, well, I guess I should go and just tell you what it is. That thing is a tRNA, which is a transfer RNA. And transfer RNAs carry something really interesting with them. They carry a specific amino acid on the end of them, on that side. And on this end of them, with the three little lines, we have something called, it's a part of the tRNA, it's not carried by the tRNA, it's called an anticodon. So anticodons have to complementary base pair with the same hydrogen bonds that you are modeling in your JMOL files for DNA with AUG. So AUG is also going to depend on this hydrogen bonding. So A goes to, and it's tRNA, so U to A to U, U to A, and G to C. And so UAC is the anticodon from which this tRNA was transferred, was brought in, and it's bringing a very specific amino acid. So we could keep going down the line and figure out the sequence. And what ends up happening is that this is one amino acid. I'm going to label this as an AA, as an amino acid. And it's going to get bonded onto, it's going to create what we call a peptide bond. I hope you remember that term. With another amino acid that gets added here, with another amino acid that gets over the top of that codon, and another one. And it'll keep going until we get this nice, and it condenses those, it's a condensation reaction, until we have this chain of amino acids that happens to then be called a protein. And that's the short story of it. So we've got rRNAs where this whole thing's happening. We've got messenger RNA, which is carrying the code from the DNA, and we've got tRNAs. But let me show you how to figure out which amino acids are being brought in. So what we have, well, I'll actually show you a model in JMOL first of this ribosome, which I think is really cool. So what I have here is that model. It's PDB file 1JGO. And what's in orange here is the RNA strand, the rRNA of the ribosome. What's in orange is the protein part of the ribosome. I'm going to pull this over here. And what we see as these things that look kind of like elephant heads with these snouts, those are the tRNAs, right? So there's 1, 2, 3 tRNAs in this ribosome. That's the most that there can be. And right on the very tip, right here, is one of the amino acids called phenylalanine. And that phenylalanine is what's going to be added onto this polypeptide chain. So if you see, there is an amino acid here, and then there's one on the end of this tRNA, and they're being added together to create this peptide bond right here. When that tRNA gives away its amino acid, it leaves the ribosome looking for another one of those specific amino acids. This green thing down here, which might be kind of hard to see, this green sequence is the mRNA sequence. So you actually would look at that and you might see those similar rings that you're used to. I just colored it all green so it would stand out against the tRNAs. But that's an mRNA sequence that's going to have hydrogen bonding between those two strands. Okay? So let me show you how to figure out which amino acid this is going to be. Let me see what I can do here. Okay, so here is your our Google Drive folder. So it's in the science folder, DNA modeling, and there's a chart here. I know it's kind of hard to see. It's called Codon Charts Table and Circle. And I double click it, and there are two things here. Let me zoom in a little bit. I'm going to show you how to read this chart first. So the sequence that we were looking for is AUG. And so the first letter of the codon is on the left side here. So this A tells me I'm somewhere in this row. The U tells me I'm in this column. So I'm somewhere in this chart. And then the third letter is actually over here on the right. So there's my G. So I know this is AUG. And the name of the amino acid is called methionine, or MET for short, or M for even shorter. Remember, there are only 20 amino acids, but there are actually 64 different combinations of codons between these four letters. So there are a lot of different options, and you'll notice that several of these give the same amino acids. So CUU, CUC, CUA, and CUG all give me leucine, but there are two more that give me leucine, UUA and UUG also give me leucine. There are 4 for proline, there are 3 for isoleucine, there are 2 for phenylalanine, 2 for cysteine. So you see a pattern, there's actually some repetition. So even if the DNA is changed and mutated, it doesn't always change the amino acid, which means that the organism isn't affected by that mutation. So what we're going to do is you're going to practice using this for some questions coming up. But here's the other version of it, which most students seem to be more familiar with. Most biochemists don't like this version simply because it's not the one that they grew up with. But this is how we start to read this one. We start in the middle and we work our way out. So AUG goes like this. Here's A, there's U, there's G, and let me scroll down. We've got methionine. There's the full name of it, methionine. Or you can abbreviate it as MET or M for short. You'll notice that there are 3 codons in here that have stop signs. UAA, UAG, or UGA. And those will all, when they get read by a ribosome, stop making the protein. They'll stop assembling amino acids in a sequence and cause the ribosome to actually fall apart and release the protein. And then the protein starts to fold in 3 dimensions based on its environment and the properties of the amino acids, chemical properties of the amino acids. And then we've got a functional protein, hopefully. Okay? So that's more or less the short end of it, but maybe we can finish coding the rest of your protein. So I'm going to write this out a little bit differently, and I'm going to let you kind of do a little bit of the work here. So if we've got a DNA sequence, I want you to actually figure out the DNA sequence by doing reverse transcription. Okay, so this is really messenger RNA, but sometimes we just call it RNA for short. And then we're going to say this is my amino acid sequence. Okay, so you're going to figure out your DNA, but the RNA sequence here is, we're going to start with AUG, CUG, GCA, UUA. Okay, so we know that. I'm going to draw my lines again because that helps me organize things. So AUG gives me methionine. I want you to find out what CUG gives you, what GCA gives you, what UUA gives you. And then do reverse transcription to your DNA. So you understand how the code from DNA affects the order of the amino acids. And sometimes one little mutation in DNA can actually change the amino acid, which changes the protein altogether. And I'm going to stop there, but I hope you understand the importance of these hydrogen bonds and the fact that they're weaker than the covalent bonds here that are holding each of the nucleotides together. All right, I'm going to stop there.